Everything's going good. Cool. We're live. Hello, everyone. Hello. Welcome to this week's stream. Got some really cool science news and articles to go over with you this week. First off, can we engineer a new and better virus? So this is a new paper coming out from one of my favorite labs, David Liu's lab. He's on the forefront of using genetic engineering and synthetic biology approaches to really change what we can do and with organisms and the different ways of expressing proteins during different conditions and targeting different organisms. And give me 1 second. All right. So this is his lab, David Liu's lab, coming out with engineered virus-like particles for efficient in vivo delivery of therapeutic proteins. I thought this was really cool because it actually allows them to use a virus-like approach to actually deliver therapeutic proteins and use the virus's specificity and their ability to target specific cells to actually deliver therapeutic proteins in this case. So this is his Twitter thread about it, and then we can go over the actual paper. So today we report in Cell as engineered virus-like particles platform that mediates efficient in vivo delivery of therapeutic proteins. In vivo delivery means, in vivo means inside, so inside the organism, including base editors. So they're looking at changing nucleotides there, and Cas9 nuclease. So Cas9 nuclease is the scissor basically of the CRISPR. So you have the guide RNA that's going to be finding specific parts of the genome, and then you have the Cas9 nuclease that can actually make those cuts, resulting in therapeutic editing in multiple mouse organs. So the in vivo delivery of proteins into mammalian cells, so we're working with mammals here specifically, remains a key challenge that limits the impact of gene editing in macromolecular therapeutics. So they're saying that the proteins, how do you deliver proteins? So if this is something where you're not just delivering genes to the organism for the organism to make it, you're specifically already making proteins, and then you're delivering those already made proteins. So that's a key difference. Once those proteins get in the system, then of course they can be doing gene editing or for therapeutics, like, well, we'll see. So virus-like particles offer a key advantage of viral and non-viral delivery, but have shown some low protein delivery efficiency in vivo. So basically the protein's not getting there for whatever reason. So in here we have like what a normal virus is, the envelope protein, the capsid protein that's protecting. These viruses package and deliver viral genetic material. So they actually have the DNA or the the DNA or the RNA, and that DNA or RNA gets delivered, and then that's what makes the protein. So for example, like SARS-CoV-2 makes the spike proteins from the RNA. So virus-like particles, capsid cargo fusion. So we have a type of protein that's delivered and bundled here instead of genetic material. My question would be, like, how do they get that in there, and how do they make sure, and how do they make sure that those proteins are what they say they are, or haven't been misfolded once that particle has been made? So inspired by previous reported virus-like protein scaffolds, we designed first-generation VLPs that package base editor ribonucleic protein. So ribonucleoprotein cargo. Ribonucleoprotein is another word for CRISPR. So ribonucleo, ribo meaning like RNA, so the guide RNA, and the protein is the guide RNA and the protein together, complex together. While V1 VL virus-like proteins efficiently edited cultured cells at high doses, they only mediated 2% editing of the mouse liver following IV injection. So a very low cutting efficiency there. So how do they change it in order to make it better? To unlock the potential of the VLPs to mediate efficient in vivo protein delivery, we systematically identified and engineered solutions to 3 major bottlenecks. Cargo release, so how those proteins are actually leaving the virus-like protein, which makes sense, right? Because if they're bound together, then how are they unbinding? Like, what's the mechanism to actually release that cargo? Cargo packaging and localization. So that would be like, how are they actually getting to and which protein are they actually targeting? It's a good question. And VLP virus-like protein component stoichiometry that reduce the potency. What do they mean by stoichiometry? Engineering component stoichiometry of engineered virus-like proteins. Oh, okay. So how much? So they're saying how much. That makes sense, because if it's not efficient, right, you could add more proteins or you can make the virus more efficient, right? So there are a couple different ways that you could actually improve that system just by having more of your protein inside the virus-like protein, or you could make the virus more efficient at delivering that protein. And maybe it would be the case that you need a very small percentage of your protein to be actually inside the particle. Maybe you need a lot. So to improve cargo release, we engineered a new linker sequence into second-generation engineered VLPs, virus-like proteins, that support efficient and release and minimize the premature cleavage of cargo before its incorporation. So, okay, so they were being cleaved before they found out that the proteins were being denatured somehow. And that would be the worst case, right? You wouldn't want to deliver something that's somehow changed and not able to do the actual job. On the other hand, you wouldn't want to deliver something at the end that's getting cleaved that isn't doing its job too. So there's 2 ways that it could be getting denatured, before and after, right? Okay. Next, we use nuclear export signals and nuclear localization signals placed on opposite sites of the cleavable linker to engineer cytoplasmic cargo localization, increasing VLP protein loading by 19 times. Wow. This is like a really common strategy. This is the right here, this little thing, it's NLS, the nuclear localization signal. So there's a lot of different little signals that you can actually add to sequences or proteins that basically tells the cell where this protein should be at any given time. So the idea is, what they're trying to do is they're trying to use that idea to put that onto what their virus like protein, to give it more of a chance to get to there, and that definitely did. There's a lot of different, the cell is very busy, right? So there's a lot of different ways and a lot of different places that these proteins need to go. And in molecular biology, it's kind of taking those ideas and that ability and then repurposing it. So a lot of molecular biology is just taking what's happening in nature and then slapping it onto whatever you want to do. You're not really reinventing the wheel, right? If there's been billions of years of evolution in order to make these nuclear localization signals, then why not just use them? So finally, we engineered an optimal EVLP, which is we engineered virus-like protein component stoichiometry that balances the amount of encapsulated cargo with amounts of viral structural proteins and protease, finding that too much or too little of either is suboptimal. I mean, that makes sense too. If you are adding too much protein, you're going to change the structure of the virus-like protein. If you're adding too little, it's not going to be efficient when it's delivered. And there's different proteases too, right, that can actually take out the protein that you want to be in there. So these proteases, I'm interested in what those are. They might be targeting proteins that would interfere with this. So they're kind of using a defensive tactic. When they get into where they need to go, it can actually be more efficient. Combining these three solutions yielded fourth generation's engineered virus-like proteins that greatly enhance protein delivery potency in mammalian cells, both in vitro and in vivo. In vitro is outside the cell, so like cellular cell culture, and in vivo inside the actual animal, typically resulting in 5 to 26 fold higher gene editing efficiency relative to, wow, that's crazy. That's amazing. What are they doing though? What are they changing? So they're changing. Okay, so they're changing some AT to GC. So one of the early differences and changes to Cas9 was basically the ability to single base pair edit. You know, you can do a bunch of different things with Cas9 now. You can actually make it so they don't cut at all, or you can make it so they make a double-stranded cut. You can make it so that they have a staggered cut, and then in this case, you're actually no cut at all, but you're changing the base pair from purine to pyrimidine, or other way around. Version 4 engineered virus-like proteins efficiently deliver base editor protein to various immortalized and primary cell types in vitro, including primary human fibroblasts and T cells. Oh, T cell, this is this is one that people have been talking about for a little bit, to actually change T cells themselves. So instead of like giving as another route to vaccination, what if we just change the T cell and put in that information directly? It's kind of like an interesting idea in order to help the immune system. We can also alter the cell type preferences, tropism, of engineered virus-like proteins by using different envelope glycoproteins. Okay, so single injections of the new engineered virus-like proteins into mice support therapeutic base editing in multiple organs. Virus-like proteins reduced serum PCSK9 levels by 78% following 63% liver editing. Yeah, go ahead. Yeah, that's fine. You can take them all. It's all good. Comparable to results achieved by state-of-the-art antiviral and non-viral delivery systems headed to the clinic. Go ahead, Siri. That's okay. That's not a problem. I'm just going to edit it out. All right. Do. Okay, no problem. All right, we're back. Okay, here we go. Single injections of that's pretty incredible. Single injections, because I mean you can think about a couple different ways of making these things delivered, right? And it could be a couple of different injections. It could be there's a lot of different ways that it could be. A single injection is pretty incredible because it could go into multiple organs. Wow. So it also targets the brain and the liver too. So therapeutically, it's incredible that it's so precise. They're obviously engineering those virus-like proteins to particles to go specifically where they want based off of the outer recognition sequences. When injected subretinally into a mouse model of genetic blindness. Wow. No way. Can they restore sight with this? That would be incredible. When injected subretinally into a mouse model of genetic blindness, engineered virus-like proteins efficiently corrected the disease-causing mutation and improved visual function. What? How did they prove that? Unlike engineered virus-like protein treated mice, lentivirus treated mice still express base editor protein even 5 weeks post-injection. Wow. So they're saying that the lentivirus wasn't as successful. Wow. In the retina. That is so cool. So they're using Western blot in order to confirm it. And electroretinogram waveform following light stimulus. Wow. Oh my gosh. So there's the wild type. There's what it normally looks like. Why can't this there we go. So here's the wild type, and then there's the untreated, just completely flatlined. But then here's the engineered virus-like proteins. It's actually coming up and being restored. Wow. Virus-like proteins engineered, giving sight. That is so cool. Because now back to it, because engineered virus-like proteins transiently deliver gene editing proteins, off-target editing from EVLPs was virtually undetected in vitro and in vivo. I guess if they're transiently, they're saying there that there's no integration into the genome. So they're delivering proteins. They're not delivering genetic material like DNA or RNA. So how could it get integrated? So a big problem there though is that if you're not integrating, how are you maintaining the proteins for a long period of time? But you could also have it integrated and then have it mutate over time, and that would be a huge problem because those mutations could cause off-target editing, and that's what they're saying is that they didn't see that at all. Because virus-like proteins are DNA-free, they should pose no risk to oncogenic DNA integration. So you could have these things causing problems where they insert. That would be a huge problem. Like, where are they actually inserting into the genome? Absolutely very cool. Our findings establish engineered virus-like proteins as promising vehicles for therapeutic macromolecule delivery that combine key advantages of both viral delivery, high efficiency, a specific tissue choice, and non-viral delivery, reduced off-targeting editing, and risk of DNA integration. That is so cool. And very impressive. Very impressive. Here's the paper. So we can go over this a little bit. So published in Cell, very prestigious journal. Highlights the engineered virus-like proteins overcome 3 bottlenecks of protein delivery. DNA-free virus-like proteins efficiently deliver gene editing proteins with minimal off-target editing. I guess how would they show that? How did they show that? Did they do like a genome-wide thing? No detectable DNA delivery from... Yeah, I mean, that makes sense. Minimized off-target editing compared to viral delivery. How did they look at those? Did they sequence? How would they sequence that and know that it's all... They must have had a random mutation on a plasmid. This is like one way that use for CRISPR that people do that. Show whether or not it's doing off-target is basically a bunch of combinations on a plasmid, and then showing whether or not it can actually bind and clip to some of those. All right. Here's the summary. We're not going to read the whole paper, but we can read the abstract. So methods to deliver gene editing agents in vivo as ribonucleoproteins, so again, CRISPR-Cas9, could offer safety advantages over ribonucleic acid delivery approaches. So nucleic acid delivery approaches like we were talking about, DNA, RNA, that's going to have problems with integration. It can mutate over time. It could not make the same protein over time. It could be silenced. It could be integrated into the genome. There are a bunch of weird problems that could happen with it. We report the development and application of engineered DNA-free virus-like particles, that efficiently package and deliver base editor or Cas9 ribonucleoproteins. By engineering VLPs to overcome cargo packaging, release, and localization bottlenecks, we developed 4th-generation engineered virus-like proteins that mediate efficient base editing in several primary mouse and human cell types. It is so cool. Using different glycoproteins in engineered virus-like proteins alters their cellular tropism. Single injections of EVLP into mice support therapeutic levels of base editing in multiple tissues, reducing serum, following 63% liver editing, and partially restoring visual function in a mouse model of genetic blindness. I think that is so cool. Restoring visual function in a mouse model of genetic blindness. I mean, that's a great example of how gene therapy could actually be a huge benefit. You know, people kind of talk about the dangers of gene editing, but man, that is such a powerful tool right there. In vivo and in vitro, so outside of the organism in cellular culture, and in vivo, off-target editing from EVLPs was virtually undetected. Okay, so it wasn't the plasmid thing. They must have been doing some type of sequence analysis. An improvement over AAV, so lentivirus, or plasmid delivery. These results establish EVLPs as promising vehicles for therapeutic macromolecule delivery that combine key advantages of both viral and non-viral delivery. So they must have been going through the figures on his Twitter, because this is what we saw before. This is their first thing. So interesting. Man, I'm jealous. This is so cool. All right. Yeah, we went through a bunch of these. These are the off-target? No. Dang. It's just so cool. I mean, it's so precise. It is so precise. How do they do that? Using FACS and GFP. These labs, man, they are so cool. How do they do this stuff? I think that the restoring sight, genetic blindness, that is such a cool way to use gene therapy. That is amazing. Wow, great paper. So the methods. It's another thing, like how do they make the particles? Production and purification. EVLPs are produced by transient transfection of Jessical producer T293 T cells. Jessical cells were seeded in T75 class the density for over 24 hours. They were transfected using jet prime transfection reagent for producing the version 1 to version 3 VLPs, a mixture of plasmid expressing VSV. Yeah, but we want the CRISPR, and there you go. And S guide RNA is for the for CRISPR. So they would have to have the RNA in there and then the protein, but how are they gonna where'd the Cas9 come from? "40 to 48 hours post-transfection, producer cell supernatant was harvested and centrifuged for 5 minutes at 500 Gs to remove cellular debris, all the lyse cells and all that kind of stuff. It was concentrated. They were injected to mice, were filtered with PBS. Weird. Okay, so yeah, high throughput. They did do next generation sequencing to check that stuff for off targets. Cell viability assay. Lentiviral production. This is their control. Jeez, man, this is such a cool paper. I am so jealous. Okay, so there we go. Amplicon sequencing for the next generation. So they did, and that makes sense. Wow. What a lab. This guy's lab is so cool. Amazing, honestly. I still don't understand. I guess they were chance of Jessical producer 293 T cells. I guess I guess I don't understand what these are. What are these? Okay, so they were transfected to make a ton of these. I still don't understand how the proteins got into the cell. What type of cell they used? The virus-like proteins. How do they make the viruses? It still doesn't make any sense to me. Well, I mean, they're probably making a company out of it. So, I guess we just won't know. Materials availability. Oh, they put it up the plasmids on Adgene. That's cool. Huh. That's crazy. They're using user cloning. That's a PCR method in order to make plasmids that have overlapping sequences that create. Yeah, you'd think that they would have just synthesized it. This is like kind of interesting that they didn't do that. I guess they're just making it and then putting it. They're transfecting it with the plasmid and having them make it inside. But how? But I guess the proteases. The plasmids must have a protease that takes down the DNA then, because then they're making it, and then because they have no detectable nucleic acids, so there's no DNA or RNA after that much time. So the plasmid must be making a DNAs. That'd be the only way that they could do it. That's a really interesting idea. Cool. Oh, man. This guy's research is so cool. I'm so jealous. All right. I'm jealous. I'm efficiently jealous. There is nothing that I'm doing in the lab that's even close to that cool, I feel like. It's like, oh, I'm so jealous. Dang. Very cool. This guy. So cool. David Liu. If you don't know about him yet, you should. You should really know about David Liu. He is incredible. All right. Next up. Next up, the dark side of online disinformation. Theoretical cosmologist. What a way to start. Spends a lot of time on Twitter. It's very popular. Science Twitter is very big. It's a lot bigger than you'd think. There are a lot of people talking about things just like David Liu, right? What we were just reading. There's a lot of people actually sharing good information on Science Twitter. It's an efficient way to communicate information. So I think that her point here is that she's found a lot of astronomy themed Twitter accounts sharing misleading or wrong information, doctored images, and sinister conspiracy theories. I mean, especially with the kind of like the reemergence, or, well, how should I say this? You know, flat Earth is like something that's actually recent. A lot of people don't realize that. A lot of people, for thousands of years, kind of knew that the Earth was round. So the idea that it's like this sinister global conspiracy was never, until recently, that was never even a thing. It never could be a thing. So I'm interested in what she's found. Wow. What in the world? That is one way. I feel like the point, here you go. I'm interested. So a lot of science. Okay, so a big question in science right now, I would say, not just this journal, but I'm just saying in general, what do we do about this misinformation, disinformation that's happening online? And, we can ignore it. We can also engage with it and debunk it. But a lot of people feel like engaging with it and debunking it is giving it more airtime than it should, spreading it, and making people more solidified in their beliefs that this is something that they should be believing in because scientists are responding to it. Because if it wasn't real, these people say, if this conspiracy wasn't real, then why would these scientists be responding to it? And I think it's an interesting question, morally, where the line is. My argument would be that scientists have already not been engaging with it and it's spreading already. And talking about the ones that have already spread, talking about the ones that have already seeped to the top is very important for science communication, to show people
why this is wrong and what it is. In that case, misinformation. Because one of the big problems that people don't understand with science is that a lot of these, the conspiracies will be at there'll be a nugget of truth to it, and then the conspiracy on top of it will be unfounded, obviously. But there's a lot of things that science it debates, and it's totally normal and it's totally healthy and fun. And that's one of the best parts about science, is that you can wonder, do I have this wrong? Is this wrong? Why is this wrong? Why am I right? And that's how science moves forward. So I think it's interesting to see in astronomy, especially something that you can see, but people don't normally have the ability to see this stuff, right? They don't have the ability to actually look at the stars and this ability. So they're kind of trusting science, right? In order to get it right. They're trusting science to make the images and give them to the public. Science, the lay public just doesn't have the ability to actually view this stuff in the way that scientists do. So how do they do this? Okay, be right back. All right. What are the most common space and physics falsehoods you've seen online? There's this one I see all the time. This is a question and answer. All right, one more time. There's this one I see all the time. Oh, that must be frustrating. Like to figure out like there's an answer to this and just see it like repeatedly misquoted. Which is that there's some giant void in space, and here's a picture of it. And it's sort of this black smudge on space. Here's what she was talking about. But that's not a void. It's a molecular cloud where gas and dust block visible light from more distant stars. There's also an image from the night sky simulation called Stellarium that claims to show Earth from the surface of Mars. But it's not. Let's see if I've seen this one. Oh my gosh. Another fake image going viral. It's a drawing someone just drew this? And it's getting, that must be so frustrating. Oh wow, that's so good. They use infrared to see it in the visible, of course. Oh my gosh, that's awesome. See, and this is the thing with conspiracies that's so frustrating, where this is just a bunch of stuff that's going to congregate and become a star. And yet that's way more interesting than whatever the conspiracy was, right? The conspiracy is that there's just an emptiness in space that we don't have no idea about. Like, it's actually going to be a star that's going to form. That is so cool. Wow, so it's a lot. I mean, obviously it's images, right? I mean, how images in biology and microbiology, we have it a lot too. With how do you make those images relatable, and so that people can actually understand what's going on, and then not take it and run with it and do their own thing and alter it. Unless it's like signed, digitally signed, I don't know, some way of keeping that integrity from the publication. We have to figure that out. It's from a simulation. There are images of really improbable skylines, like a sunset with a full moon next to it, which is not possible. There are a lot of images that get cycled around. There's Twitter accounts that basically track those and point them out in real time. Wow. Well, there you go. That's nuts. I'm sure people actually... Just another... Let's look at some of these. This is funny. Just another daytime photo with a too big moon pasted into the background in front of the clouds. Yeah. That is kind of weird. Yeah, man. Amazing. I love it. Blood moon. Like, no, dude. No. The moon is not full. Oh my gosh. It's a stock photo. It's just a stock photo. Like, why can't people just say what it is and it's art? That's fine. If it's art, then that's cool. Why say that this is, something new? And I don't understand why people have to lie like that. What's the weirdest claim we've ever seen? All of them are so weird. There have been a couple of claims going around about the idea on a certain date Mars is going to appear as big as the moon in the sky. One went around a while ago that there was going to be an alignment of planets that was going to change gravity on Earth. It was going to briefly make us all lighter. How do people believe this? Oh my gosh. At least they're engaging with it. I mean, if they have a Twitter account, talking about the falsehoods, the worst thing that can happen in a scenario like with that, right, is that someone actually takes that and is like, oh, this is a whole Twitter feed of cool things that are happening. On the face of it, it seems frustrating, but not necessarily as troubling as health misinformation. So they're talking about all the COVID misinformation that's going around. Answer: the way it goes is the way you see things that's false and you believe it, right? Some physics or astronomy thing that is false, or for whatever reason you think it's true, and then you discover that it's not an accepted idea. All the experts say it's wrong, but then at that point, either you accept what the experts say, or if you still think that thing is true, then the experts are lying. And if the experts are lying, then that leads you directly to some conspiracies, right? Because you have to say, what else are they lying about? Why are they lying? Who are the people running the show? Exactly. I mean, the idea that the entire scientific community could ever be aligned to, even if like they just don't. It's so hard to explain, but take the distrust that you have with your own boss at your job and just multiply that by all of the labs that exist and all these people have fact checking their boss in real time. Science is not a monolith. It is not. There's a lot of people that believe a lot of different things in the scientific community, and there's gonna be a lot of whistleblowers that would just not allow that to happen at all. That happens with things like the flat earth conspiracy, which is not something you can believe in if you don't believe that everyone involved in the space community is a liar. I mean, the flat earth conspiracy is one of those absolute game-breaking, mind-melting conspiracies, right? Where if you believe in the flat earth, there's literally there's nothing else that you can believe from any other trusted source, because space is fake. All the entire scientific community is fake. It's like, I mean, and there's like, gosh, there had to have to be hundreds of, like 100 million people involved. I mean, just imagine that level of coordination. It is just be impossible. And yeah, I mean, it's so hard to believe. And like, why hasn't anyone live streamed going to the edge of the earth? If the flat earth is real, right? Why hasn't anyone just walked out there? What an amazing event. Live streamed going to the edge of the earth and just looking down. I don't understand. So back to the article. And if they're lying, they must be coordinated by some high up conspiracy, and then that leads into all the other conspiracies that require some kind of global leader. Yeah, it's the NWO kind of thing. Once you dig down into it, they all kind of look the same. They get you to paranoid conspiracies, anti-Semitism, the elites are holding us down kind of thing. Absolutely. That's the NWO, lizard people are running the show kind of ideas that are just, you got to feel for those people, because the world that they live in is, just imagine every single corporation, every single government, every single scientist all working together in order to keep you down kind of thing. And that's just like, it's really sad, to think about that these people have constructed this this huge enemy that's so much bigger than them, that they could never win against, that they could never progress against, and it's just happening to them. And I really feel for those people, to be honest with you. That's one of these things that ends up being really dangerous about blindly accepting that someone just doesn't believe in gravity. If you get invested enough in that falsehood, you have to get to a point where you're believing a genuine conspiracy. So I think it's really troubling when things that are just false are propagated and shared and uncritically believed on a wide scale, because a small fraction of those are willing to end up falling down a rabbit hole. What do you think that leads people to share false information in the first place? I think a lot of people want to try to make sense of the world, and it's, conspiracies are one of those things where it's convenient. It's easy to believe that everything is this one answer, whether or not it's the NWO, whether or not it's, the whatever organization that needs to be. It's a very alluring to have that one answer. In reality, a lot of these things, it's hard to know what's going on. You got to do a lot of your own research. And that's one of those reasons why that term has been co-opted, is because it's hard. It's hard to do it. So people want to tell someone that they've done their research. And it's, a lot of times just Googling something or reading Facebook is not going to give you the right answer. And they'll think it's the right answer because it's just 200 times pasted the same thing, the same narrative. And they say, well, I've seen this 200 times, so how can it be false? Like, how can all these other 200, posts be fake? And, unfortunately, they're trusting other people. They're trusting the sources, the original source. And what this person's talking about is that they're being taken advantage of. They're having misplaced trust, and yeah, it's really unfortunate. And I don't know how social media, I don't know how the internet, how we get past this point, but there has to be something where people start believing in expertise again, that people believe that like there's this astronomer has a good faith effort in order to try to figure out what's going on, and they're thinking about this stuff a lot. They're thinking about this all day. And they're not going to be lured into these honeypots that are just so easy to disprove. So anyway, back to the article. So what do you think that leads people to share false information in the first place? Answer: I think a lot of times when people share false information about science, they don't know it's false. I agree. They're mistrusting. A lot of people don't have the skills to really evaluate the sources that they're looking at, or really don't care. They share a video because it's cool and they don't check where the video comes from. I definitely agree. They don't check to see that a bunch of people are replying to the original tweet saying it's fake. They don't Google it. Most of the time, I think people share stuff because they think it's neat and they don't know that it's wrong. I agree. I think a lot of people, my problem isn't with people that are misinformed. It's the people that are purposefully making that stuff in the beginning. So like what we were just talking about, who edits that photo in order to make it look fake, make it look real, and not just say that it's art. It's that person. That person is purposely making disinformation, and it's so dangerous. I don't understand, other than like a laugh or like some type of like, oh wow, I really fooled a lot of people. I mean, what's the point? I don't get it. Back to the article. So when people get more invested, it happens the way that all conspiracies happen, which is the people get into the idea of secret information. Yeah, it's very alluring, right? The appeal of conspiracy theories is that there's some secret explanation that you can have access to that most people don't have access to. I think this is right. It's like and normally the conspiracy is so easy and it wraps everything up, and it's just like, that's why, so that's a big part of the flat earth stuff and the moon landing hoax. All these kind of ideas come down to the appeal that conspiracy theories have. It's easy for people to get invested when they think they found this amazing secret thing. And it's, yeah, exactly. Like that you don't have to, you circumvent all the knowledge that you need to acquire in order to get a PhD in astronomy when you just think the flat earth is flat. They got it all wrong, man. They don't want to end up with the mundane explanation that actually their physics books were correct, and nobody finds that exciting. It's very true, unfortunately. As an expert with a big platform, what are the personal strategies for dealing with space misinformation? So I'm interested to hear, like, in biology, we're trying to take a crack at it with, the COVID misinformation. So what are they doing with the space stuff? I don't put a lot of effort into debunking because I don't know how effective that is. And it's just really thankless. It's so true. It's not nobody's going, oh, thanks for showing me that it was the thing that everyone thought all along. It's so true. The people who are really invested just yell at you all the time, and everyone else is like, why are you spoiling their fun? There's no happy ending. No, that is so frustrating, but it's true. There are some studies that show that if you talk about the wrong thing, even in the context of debunking, people remember the wrong thing. I've heard that as well. It's really hard to find ways to present the wrong information in a way that doesn't somehow reinforce it. Yeah, I've heard that as well. Even like for my own self, like I remember, like, for example, I remember Mythbusters episodes, but I don't remember if it's been busted or not all the time. Just thinking about that. I love Mythbusters. I was watching it all the time, but do I know the outcome of every episode, whether or not it was false or true or myth or whatever? Like, I can't remember that. And sometimes the story, I forget how it unraveled. There's also the psychology of showing something someone something, and then they actually you show them why they're false, and then they create a even stronger conviction that their wrong idea is correct. I think that's all too apparent with a lot of the COVID and anti-vax misinformation. What I try to do is share the real information. I try to share it in a non-judgmental way. When people ask me a question and they have a misunderstanding, I try to correct the misunderstanding in a way that doesn't imply that they're dumb to believe it in the first place. I try to start from the point of view of, it's great you're interested in learning more about this. Here's how you can learn more about it. It's really tough because a lot of times people really do mean well and they just want to share the science, and people get defensive when you tell them that what they're sharing is wrong. I agree with that. I think that there's a lot of people that, if you're interested and you're reading blogs or you're reading COVID information or anti-vaccine stuff about vaccine safety and all that kind of stuff for, literally months or maybe, over a year now, like what, like get into the science, right? Like that's what I don't understand. Like get into it, get into figuring out what's going on, and then try to improve vaccines. If you don't think the vaccines are safe, then make a safer vaccine. I don't understand the people that spend so much of their lives on researching a subject, but then don't do the good work to actually go a step farther and research it themselves. Actually do research and contribute to the research themselves. It calls into question how good of faith that there are in general, because if you really thought that vaccines were that fake or that dangerous, then why wouldn't you try to make them better, right? That's the thing that I don't understand. To the article. If a person does just want to share cool science, what useful fact-checking strategies do you have? Answer. The main thing is to know where it came from. Yeah, I mean, for me, it's a lot of journals, right? Going straight to a journal source and just talking and looking at the primary source of information. A lot of people, it's a skill that a lot of people don't have nowadays, is to actually read primary information, and by that I mean the original source, not the news article about the science study, but the study itself. And that's what a lot of people don't go to. They go to the news article hoping for like a layman's explanation, but in reality, they just get misinformed sometimes. News people or journalists will try to spin words. They'll try to emphasize things that aren't, they were a gray area, but now it's black and white kind of thing. So it's always good to try to go for the original source if you can. And on top of that, what's the best place to read in an academic paper then? Well, it's the title and the abstract. You can, the abstract's usually written in the language that you can understand. If you can't understand it, then, Google it, Google the terms, read, what's a ribonucleic protein kind of thing, ribonucleoprotein. So Google that, see what that is. And then you learn that term. That's the best thing I think of. Back to the article. A lot of people share things from fake sciencey meme accounts called like Amazing Physics. And the accounts just scrape images and videos from other sites. Oops, excuse me. They don't have the credit on any of the content they put up, and they just link to the real information about the content they put out. And they don't link to the real information about the content they put out. They share the image or video. The image or video might not be totally fake. It might be something completely misinterpreted. I really encourage people never to share that stuff. Never share a video that has no attribution, because you don't know where it came from. If you can't find out where it came from, you should not share it. That would cut down like 80% of the bad physics and astronomy stuff out there. There's something amazingly surprising, always be suspicious. Yeah. So if it's too good to be true, it probably is. So, it's, sometimes, it pays to be skeptical. And that's sad. it's sad that the information on the internet is more and more turning into conspiracy and misinformation. But hopefully there are good sources of information out there. Hopefully there are good places that you can go to trust primary information and actually reading primary information rather than a share of a share of a share, an image that has things circled and drawn and all this kind of stuff, when it's like, yeah, you can just share the original one and it'll be fine. So hopefully this YouTube channel is that for you, and you can trust what comes out here. That's my goal. That's my goal, to make science approachable and science interesting, so we can talk about the cool stuff that's going on about science without neutering it, without clipping it down to 10 seconds. So, and all of the actual content and the depth is taken out. And I think that's kind of like what's missing right now in this space and YouTube. And hopefully this gives that space to you. All right. Next up. All right, next up. World's nations finally hammer out the world needs to deal with its plastic problem. Hopefully we can start hammering out a global treaty on plastic. The world has a plastic problem, and hopefully this treaty can help stymie some of the plastic pollution that's happening right now, because it is bad. There is so much of plastic waste that's in the ocean right now that is crazy, between the Pacific Gyre, the huge garbage patch in the ocean. Plastic is just sticking around for so long, and it's existing plastic. So it's already in there. This can't be necessarily recycled, a lot of this. So how are we going to deal with it? How are we going to clean it up? Here we go. So you have a treaty, maybe. Because I'd heard that a lot of the plastic waste actually comes from a small number of sources. So maybe we could start stemming that, and that would have a huge tangible impact. So each year, an estimated 11 million tons, of plastic waste enter the ocean, equivalent to a cargo ship's worth every day. Oh my gosh. The rising tide in the oceans and beyond is just a symptom of such wider problems. Unsustainable product design. For real, why are things lasting so long, and why are we constantly reusing these things? Short-sighted consumption. You get something from a fast food restaurant and they give you something that's made of styrofoam that's going to last on the world for 1,000 years. I don't get it. I would pay. I should have the option, right? Give me the option. I'll pay $1 more to have that sustainable option. And insufficient waste management. Yeah, a lot of it is just a lot of people don't have the ability or the waste drop off places. It's like there's no place for it to go. To curb the flood, says Jenna Jambek, an environmental engineer at the University of Georgia, we need to take more action. It needs to be further upstream in the production process. So true. That's exactly what negotiators from 193 countries are setting out to do when they meet in Nairobi, Kenya next week. This ambitious goal to create a negotiating committee that will hammer out within 2 years a new global treaty intended to curb plastic pollution. An already released proposal modeled on the UN's climate treaty would have nations adopt action plans, set binding waste reduction targets, and establish monitoring systems in a new global scientific study advisory board. Quote, "It's about time," says Chelsea Rockman, an ecologist at the University of Toronto, who has called on nations to tackle the issue. Yeah, I mean, it is bad. How are they going to do this, though? The monitoring is an interesting idea. Would they use satellites? Many countries allow companies to keep such numbers private. So of course, they're not. The Ocean Conservancy, for example, has struggled to find out how many recycled plastic firms are using. Wow. Researchers are still pondering which numbers would be the most useful and how the treaty might help make that information more available. Oh, okay. Here we go. This is what I was talking about. Remote sensing via satellites and drones, for example, could more easily identify plastic pollution trends, reducing the need for labor-intensive ground surveys. Yeah, ground surveys for the entire world? I mean, that's going to be... Sheesh, that's bad. I mean, satellites, some kind of remote sensing is definitely going to be needed. I wonder if there's like a specific wavelength that could be used in order to like specifically look at plastic, like the bonds within plastic that would be different than what's found in nature. So maybe we could actually use off-target wavelength monitoring. How much plastic pollution is too much? It's clear that plastic bags, discarded fishing gear, and microplastics can kill wildlife, but scientists are just beginning to figure out how to calculate those risks. That's an interesting question. It'll be challenging in the short term to stop using virgin plastic. Yeah. A big reason is that many uses of plastic are seen as essential. Single-use plastic items are common in healthcare, for example, to prevent contamination and infection. That is a huge thing for me personally, actually, in the lab. So, there's so many times in the lab where we're using something that's plastic, using it once because it's sterile, and then throwing it away. And here I am trying to devote my life in order to make this problem better through minimize the human impact on the environment with my own research. And here I am having to use plastic a lot in order to do this. Glove waste, PCR plates, tips. Tips are a big one. When you're doing molecular biology, you're doing pipette tips, and you're changing the tips after every single use. And it's one of those things where you're actually encouraged to change your tips. I mean, you don't want to use the same tip because you could make the entire experiment contaminated. And if you do that, then you got to do the whole thing again. And so it is such an interesting problem in science, is the renewable use of these materials, and actually figuring out how science can be more renewable as well. I think about when I was an undergrad. So, like a little quick anecdote. My then girlfriend, now wife, we made a pact to never buy paper towels. Just never buy it. We have never bought a paper towel as a couple, and I wanted to do something like that with the lab, right? With the lab that I was going in. So I come in as an undergrad, and I'm like, these paper towels, man, we should never buy paper towels. We should just get a regular towel, a reusable towel like we do at home, and we can wash our hands with that, all that kind of stuff. And everyone's just like, no. Are you crazy? It's going to cause such a contamination risk. Who's going to wash the towels? Who's going to wash the hand towels? At home, it's like we can easily throw it into the laundry and it's not that big of a deal. But, in the lab, it's this whole thing, like washing the towels. And it's kind of it was just instantly rejected for good reason. And I still don't do it, to be honest with you, at the lab, because it's just not feasible for those reasons. Do you want a contamination risk, again, that could impact the entire lab's research? Like, probably not. It's not the best. And so I just made the personal decision to only have that done for me at at my house. But it's yeah, it's a weird problem. How can science be more renewable as well? It's a really good question. Hopefully we start curbing our plastic emissions, though. Our plastic waste is a huge problem for the earth. So cut down. Figure out how you can cut down on your plastic waste as well. Okay. How can we, as scientists, engineer plants to fight climate change? So, in this article, we have a biotech firm that's going to genetically engineer a poplar tree to grow really quickly and suck up a lot of carbon. I think that's a really cool idea. A California biotech company seeks to create fast-growing trees that can rapidly soak up atmospheric carbon dioxide, has announced its first experimental results. The firm's genetically enhanced poplars grew more than 1.5 times faster than unmodified ones in lab trials. That's cool. That's a good idea. I mean, as CO2 increases in the atmosphere, well, having trees that can actually take advantage of that and soak up even more carbon and bring it down would be a great idea. It's a great first step, says Sophie Young, a plant biologist of Lancaster University who is not involved in the work. How do they find this person? They just randomly ask someone. But she adds, there's a huge big caveat. The trees grew in a carefully controlled greenhouse as opposed to outdoors. Well, I mean, I don't think that's like a huge problem, but, we'll see. Scientists and environmentalists have promoted tree planting as a promising and easily expendable way to draw down atmospheric levels of carbon dioxide, the primary cause of global warming. The trees, which are roughly half carbon by dry weight, absorb the gas from the air and turn it into stable forms of carbon, such as wood and roots. It'd be interesting. Poplar, I guess you could use that for building and stuff too. Poplar is a good wood, right? But how fast the trees soak up carbon is limited by numerous factors. What's rubisco? Rubisco is the biochemical protein that brings those carbon dioxides into sugars. How do they do that? How do they make it do that? I wonder what. To sidestep the problem, researchers from a company called Living Carbon used a bacterium to insert genes from a pumpkin and green algae into poplar trees. The foreign genes enabled the trees to have a lower photorespiration rate and to recycle carbon from the phosphoglycolate into sugars essential to growth. Wow. Other research previously engineered similar traits into tobacco plants, but to my knowledge, the approach has never been tested in a tree before. So they're not doing carbon concentrating mechanisms. They must have been doing a new rubisco, maybe? How are they doing that? Living Carbon, founded in 2019, is growing the engineered poplars inside a converted music recording studio in San Francisco. All right. As someone who spends my time in Zoom, it's a very nice. Oh my, what? It's a preprint server. It doesn't. That's a weird. That's not really a good way to call it. The company reports its study posted today in the preprint server bioRxiv, which does not require formal peer review. It's kind of like a Google Drive for publications. It's not so much that it doesn't require it; it's that it's a preprint server before publication, so it does. It's not peer reviewed. Incorporating a photorespiration bypass in C3 plants has shown promising results of increased biomass and grain yield. Despite their globally dominant role in atmospheric carbon flux, the drawdown rates of most trees are currently limited by their C3 photosynthetic metabolism, and efforts to improve the photosynthetic capacity of trees, such as reducing energy loss and photorespiration, are currently lacking. Here, we selected a photorespiration bypass pathway and tested its effectiveness on photosynthetic enhancement in hybrid poplars. Wow, that's cool. The design includes an RNAi strategy to reduce the transportation of the photorespiration product, glycolate, out of the chloroplast, and a shunt pathway to metabolize the retained glycolate back to CO2 for fixation through the Calvin-Benson cycle. Cool. That's the molecular mechanism there. That's cool. So they're doing a bypass pathway, and they're making it. They're pushing it there with RNAi, and RNAi is RNA interference. So they're interfering with the normal route that it's going through and pushing it through the other route that they have through RNAi. The numbers are encouraging, but not overwhelming. Yeah. The tobacco enhancement project. Yeah, tobacco is used a lot for a lot of botany research. Interesting. Cool. We need better plants. How can we evolve plants quicker? We need plants that are going to be doing better in high CO2 environments. We need plants that are going to be doing better in high temperature environments. And we also need plants that can just take down more CO2. So this is an interesting study. If you wanted to check this out, TAO Tau 2022. Good research. All right. All right. What does DNA tell us about human origins? So next up, we have oldest human DNA from Africa reveals complex migrations. Africa is the birthplace of our species. But hey, you don't know what I am. Well, our species? Our species? But ancient DNA from the continent has so provided relatively few clues to our history there, partly because researchers have struggled to recover genetic samples that survived the hot, humid climate in Africa. Now, an analysis of ancient DNA from 6 individuals from southeastern Africa offers a glimpse of the lives, moments, and relations of people who occupied the continent between 18 to 5,000 years ago. What? Ancient DNA. Where are this ancient DNA coming from? Where is it coming from? Does anyone know? Let's see. multiple lines of genetic and archaeological evidence suggest that there are major demographic changes in the terminal late Pleistocene epoch and early Holocene epoch of sub-Saharan Africa. Inferences about this period are challenging to make because demographic shifts in the past 5,000 years have obscured the structures of more ancient populations. Here we present a genome-wide ancient DNA data from 6 individuals from Eastern and South Africa spanning the past approximately 18,000 years. Wow. Doubling the time depth of sub-Saharan African ancient DNA. Increase the quality of 15 previously published ancient individuals and analyses, and analyze these along with data from 13 other published ancient individuals. Wow. So this is a huge genome like a genome-wide. They put in a bunch of different people. Wow. Distribution and main ancestry components. Let's see these figures. Whoop. Let's try this. Wow. How are they able to do this? That is incredible. How are they? That is so cool. I'm blown away by that. How in the world? Wow. Considered in that context, in African artifacts of that period, we've seen a ton of hints that people are connecting in different ways. He says mixing and matching artifacts from different places. Considering in that context, the new DNA data suggests that sometime after 2,000 years ago, ancient Africans stayed closer to home. Stone tools found in their rock shelters take on a local flair, and their DNA suggests that starting around this time, people traveled shorter distance to find mates. Whoa. Huh. The 6 new genomes are a welcome addition to Africa's sparse record of ancient DNA. That's so interesting. You'd think that there'd be a lot more. But she's not swayed by the team's ideas what happened about 2,000 years ago. There's a lot of assumptions in that analysis, she says, and it's not clear to her that the authors considered alternative explanations. Ooh. How could you so... Why would she's not convinced. The distinct genetic signatures of those ancestral populations indicate they were mostly isolated from one another for vast amounts of time before eventually coming together, said David Reich, a population geneticist at Harvard Medical School who co-led the study. If you look at Europeans and East Asians, maybe they're separated by 40 to 50,000 years, he said. These 3 groups were basically separated 200,000 years ago, then come back together maybe 80 or 50,000 years ago. Wow. Yeah, I mean, Homo sapiens is a lot older, as a species than people really think or take note for. The range is also only an estimate. We are learning a lot about our species still, and it is so cool that we can still have so much to learn. Very cool. I'm really interested too about other sapiens that lived in other hominid species that also lived during the same time as us, and where they went. Because we had more than a dozen hominid species living on the earth at the same time, and now there's only one. So it is a very weird thing for me. I would love to know more about that, and for science to tell us more what happened there. All right. All right. All right. I think we got time for one more. Let's do one more. Microbes are the future. They are. It's just you can't say no to that, because microbes are just gonna make everything for us in the future, from plastics to fuel to our food. They're just gonna happen. It's just gonna happen. You can't do anything about it. I'm sorry. You can't change it, because they're very efficient at things, and we are figuring out ways to make them even more efficient and better and make new things that they've never made before. And here, microbes convert industrial waste gases into commodity chemicals. So what if we can actually use the byproducts that we don't even use, that's considered waste, and actually use them for useful things? Enter microbiology. Chemicals cost more than just money. Today, petrochemical production spews out 2% of the world's greenhouse gas emissions. Now, researchers have taken an important step to vastly reduce that footprint by using bacteria and waste gases from steel plants rather than petroleum as the starting ingredient for dozens of commodity chemicals. That is such a cool idea. I mean, for chemistry, you can start in a lot of different ways along the process. So this is a way to start with something that's a little bit more bio-friendly. Because researchers may be able to expand it to others, it could help the chemical industry escape its reliance on fossil fuels and effectively remove carbon from the skies. Oh, what a great intro. Man, that's cool. Harnessing biology to utilize waste gas, and produce industrial chemicals is really exciting. A biofuels expert with the Lawrence Berkeley National Laboratory. It goes after two sectors at once that are difficult to decarbonize: steel production and industrial chemicals. Tackles a very hard problem. Humans have exploited microbes for thousands of years to make products like alcohol, cheese, and yogurt. It's true. Alcohol, cheese, yogurt, it's just food that microbes have already eaten, and we're eating the microbe waste. That's what alcohol is. That's what cheese and yogurt is. They've already grown in there, and we're eating it. We're pre-digesting our food with microbes. It blows my mind. But industrial scale biotechnology didn't really take off until the beginning of the 1900s, when, for example, chemist Chaim Weizmann engineered several species of Clostridium and bacteria to convert starches and sugars into acetone, an essential ingredient for making cordite, a smokeless alternative to gunpowder. Weizmann's success was a key to the Allied victory in World War I and gained him fame. A Zionist politician as well as a chemist, Zeinman eventually became Israel's first president in 1949. Wow, look at that. I had no idea that Israel's first president was a scientist. That's really cool. With the rise of petroleum industry in the 1950s and 1960s, how can they gave chemical manufacturers cheaper starting materials for dozens of commodity chemicals, regulating most microbes to the industrial waste bin. I mean, this is like, right? Plastics. I mean, I was laughing at it because it's like, it seems ridiculous, but then you think about everything that's coming from that, right? You got the huge expansion into fossil fuels, the mining that happens there, and then you have the plastic boom, right? Everything starts being made from plastic. You know, it's just, yeah, it is what a sad time. It's hard to imagine, though, so much of our world without that happening. Even how I'm speaking to you now, you know, it has everything to do with silicon chips. And it'd be a different world if plastics weren't invented. That's for sure. But not all. Today, yeast and Escherichia coli are widely used to produce a range of commodity chemicals, including ethanol for fuel and compounds used to make pharmaceuticals and plastics. But these industrial workhorses come with their own environmental footprint because the sugars and starches they ferment come from crops like corn and sugarcane. Dude, like, By 2026, corn grown to make ethanol in the United States will take up to 19% of the nation's farmland. Oh my gosh. Why? I mean, this is a weird issue too, because there's the bill that was passed by President Clinton that requires us to actually have 10% of our fuel be ethanol. So you have all these different starches and sugars coming from corn. Oh, that's so sad. Now, synthetic biologists are pushing to grow microbes on a more environmentally friendly diet using waste gases produced from industry, garbage dumps, and crop production, says Michael Kopke, synthetic biology at LanzaTech, a biotechnology company. In the 1990s, researchers engineered Clostridium autoethangium, a bacterium originally enriched from rabbit feces, to produce ethanol from hydrogen gas and carbon monoxide. What is this? Getting some spam in the chat. Oh God. Look at all this spam. Gonna have to ban some people. Gonna have to start banning people. All right. I know if there's anyone that has any experience with biofuels or any microbes or anything like that, that'd be so nice. In June 2018, LanzaTech opened its first production plant that used the bacterium to make ethanol from a steel mill's waste gas. A mix of mostly carbon dioxide and hydrogen gas would otherwise be vented into the atmosphere. The company now uses this technology to make 90,000 tons of ethanol per year. Jeez. Wow. Still, the bacterium has never matched the versatility of E. coli or yeast. Up to now, engineering Clostridium has been just hard, says Michael Jewett, a synthetic biology at Northwestern University. The bacteria are slow growing and they die from exposure to oxygen. In addition, researchers have few tailored gene edited tools to alter the microbe's metabolism. That's interesting. I mean, you would think that all the gene editing tools would be able to edit it. Why? How do they? Is it just tailored for that one organism? I don't understand. Not anymore. I love that. Jewett, Kopke, and other colleagues use a multi-step strategy to coax C-Auto into churning out acetone and isopropanol, a colorless liquid found in disinfectants and cleaners. They started with a collection of 272 sugar-eating Clostridium strains maintained by retired South African microbiologist David Jones. The research team broke apart the bacterial cells and mined them for enzymes that appeared to play a role in the acetone biosynthesis. They used proteomics in order to see which ones were able to do it. I wonder if they had predicted which ones would be better, or if they just took all of them and then used like kind of an erased. Nope, they sequenced the genes for 30 of these enzymes and inserted combinations of them into C-Auto via gene delivery vehicles like plasmids. Okay, so they're using like a directed evolution approach. Oh, cool. Nice. That's awesome, man. Ultimately, Jewett, Kopke, and other colleagues generated and screened 247 genetic variants of the bacteria to find the best chemical producers. The resulting report out today in Nature Biotechnology are C-Auto strains that continuously convert steel waste gases into enough acetone and IPA to make them viable candidates for large-scale commercial production. That is so cool. Yeah, directed evolution. If you can use nature's diversity and then screen that diversity, then we could try to create a product that is just better. Jewett expects that some processes, Jewett expects the same process could make the bacteria produce a variety of other chemicals such as butanol used in varnishes and propanediol found in cosmetics. Scown adds that novel synthetic biology techniques would also help transform other recalcitrant microbes into industrial workforces. We're just scratching the surface in terms of other microbial hosts that might be viable in the future. I mean, considering that 99% of microbes are not transformable, not able to be genetically modified, or even cultured in the lab, I think that there's a huge host of tons of species, millions of species of microbes, to be honest, that could actually make a big difference in the future that we have. The advance could also open up the door to genetic engineering microbes capable of feeding on other gases, such as those produced by municipal solid waste and agricultural debris. And if, like Ciato, these microbes are use more carbon in making their products than release into the atmosphere, industrial chemical production may someday transform from a greenhouse gas rogue into a champion. Well, let's check out this paper. Very cool. Carbon negative production of acetone and isopropanol by gas fermentation at industrial pilot scale. Wow. That's huge. Let's hear all the authors. Very cool. Many industrial chemicals are produced from fossil fuel resources. Many industrial chemicals that are produced from fossil resources could be manufactured more sustainably through fermentation. Here we describe the development of a carbon-negative fermentation route to producing the industrial important chemicals acetone and isopropanol from abundant, low-cost waste gas feedstocks such as industrial emissions and syngas. Using a combinatorial pathway library approach. Okay. So they use different parts, combine them together. And the library is all the different types of mutation and the different types of pathways combining together in order to make that product. We first mined a historical industrial strain collection from superior enzymes that we use to engineer the autotrophic acetogen, Clostridium, autoethanogenome. Next. All right. And 1 second. Next, we used omics analysis, kinetic modeling, and cell-free prototyping to optimize flux. Finally, we scaled up our optimized strains from continuous production at rates of 3 grams per liter per hour and 90% selectivity. Life cycle analysis confirmed a negative carbon footprint for the products. That's incredible. Unlike traditional production processes, which results in the release of greenhouse gases, our process fixes carbon. These results show that engineered acetogens enable sustainable, high-efficiency, high-selectivity chemical production. We expect that our approach can be readily adapted to a wide range of commodity chemicals. That's incredible. Let's see their figures real quick. So the pathway optimization, the genome mining of everything, combining multiple of these. So they're different, multiple pathways with multiple different types of mutations in each pathway, each probed on each of, with every combination. That's pretty incredible. How are they able to have such a high rate of library efficiency going through a majority of their library if they didn't have those genetic engineering tools like they needed before? Cell optimization, the cell-free prototyping, omics. metabolic modeling. So that's to make sure that they're making what they're thinking. The cell-free prototyping is an interesting thing, because they're just looking at the products of each of these pathways, I guess. And then they're scaling up to the process optimization, the fermentation and development, scale up, and then finally the life cycle analysis. Wow. Look at those, the acetone. They're all pretty good. Above the reference strains. That's incredible. Wow. That is really cool. Strain optimization, so you have the different pathways working in order to make the products. How did they know that each of their pathways, mutants, and combinations were going to be making their desired product? I guess that's how they're making the mutations here, and then they're doing that here. They're looking at the different pathways to see what's being made and making sure that each of them are being made along that way, that biochemistry. Because when you have so many different pathways here, you could have a lot of the resources being made and, shunted towards lactate or something at the end here. And that's not what you would want. You don't want like lactate being where all your energy is going. So you'd have some type of flux monitoring. How are they doing that? I don't know. Process optimization. And then finally the life cycle analysis. So there you go. That's it. Microbes are the future. There's no stopping it. It's going to happen. Lou, 2022. Microbes. They're cool. All right. So that's going to be the live stream for this week. Hope you have a good week. Catch me tomorrow on Ministry of Science on Homozygotes Twitch stream, and then on Sunday, the Philosophy of Science with Dr. Bwin Bwin. Going to be going over panels on both of those days. This channel is going to be having more videos that are going to be coming out. It's all going to be really fun coming up. So share the link, subscribe, like, all that stuff. You know what to do. And I'll see you next week. Wednesdays, 9 p.m. Having fun, going over some cool stuff in science, making it relatable, making it fun. What more can you want? I don't know. I'm trying. We'll see. If you have a good science article that you think is interesting, drop it in the Discord. You can leave a comment during the live stream, and I'll go over it. All right. Well, have a good week.